Okay, we'll go ahead and get started. Welcome, everybody. Thank you for joining the Release 41 EuPathDB webinar. My name is Omar Harb, and I'm part of the outreach team at EuPathDB. I'll be joined today by Evelina Bashenko, who and Suzanne Warrenfeldt, both part of the outreach team at EuPathDB. Evelina specializes in fungal biology and is based in the University of Liverpool. And Suzanne specializes in multiple organisms, part of EuPathDB, and is based at the University of Georgia, and I am based at the University of Pennsylvania, and we're all part of the EuPathDB project. So today's webinar. So before I get started with we get started with the webinar, some housekeeping rules. So in GoToMeeting or GoToWebinar, if you have questions at any time during the webinar, feel free to use the question section or the chat section in your GoToWebinar window. You can just type questions in there. One of us will be monitoring the questions at all times, and we will either answer them by typing a response in there if it's a quick response, or we will answer them verbally as we get a chance. In addition, feel free to you can raise your hand as well if you have a question that you want to actually voice it verbally, not write it, and we're happy to turn on your or unmute you and allow you to ask the question. Also during this webinar, of course, if you have any suggestions or comments about the databases in general, we are always happy to hear those and happy to discuss them as well. Okay, so as I mentioned, today's webinar is specifically, the goal of today's webinar is specifically to cover the various data sets and functionality that were made available in the last release of EuPathDB, which happened in December, right before the break or the holidays in December. And one of the quickest ways for you to find out what is new in any of the databases is if you go to the EuPathDB homepage, eupathdb.org, you can go down to this news and tweet section right here, and there's a section or a little link here called All EuPathDB News. And once you click on this, this will take you to a page that collates all of the news from all of our databases. So as you see here, the top one is AmoebaDB, and as you scroll down, you will see that there are news items from all of our databases highlighting the various data sets and functionality that were loaded. Just as a reminder, since EuPathDB sites are all built on the same infrastructure, basically, if we make a tool available or design it because it was a request from, let's say, the Plasmodium community, that same tool, if it makes sense for other resources, will be made available in all of the resources. And so that's sort of a nice functionality that can be applied to everybody. So the way we're going to organize the webinar today is each of us, myself, Eve, and Suzanne, will take turns covering and highlighting various data sets that came out and were made available. Just again, and I will cover this at the end as well. If we have a little bit of time, I will take some time to highlight some data sets that are coming in our next release, which is expected towards the end of February. And I will cover again sort of the process that we take when we the decision that the process we take in deciding which data sets get loaded and get prioritized. But just briefly to let you know that we do hear about data sets from multiple sources, including from people like you. You send us an email to the Contact Us link at the top of or the bottom of any of our pages and tell us about a data set, either your data set or a data set that you've just read about and you think would be useful to include in one of our resources for the community. The other way, obviously, we do regularly look through publications and try and determine which data sets are are useful for a resource like EuPathDB. And then we have advisory committees for most of our databases, and those are basically people who work on those organisms. And if you go to any of our databases, you can click on the about the database, and you can scroll down and look at the advisory groups. So we have scientific advisory teams, and you can see who's on those advisory teams. But they're typically primary investigators who work on the organisms for that database. And they help us prioritize these data sets and give us an idea of which one, if we need to decide which one to load first, they give us some input about that, and that's incredibly helpful. The first thing we're going to cover today is I will highlight a couple of new features that were, or tools that were loaded, and those are specifically two new gene page tools. One of them is an ability to run an InterPro Scan on your gene of interest or protein of interest. And the second one is to run a GPI Anchor Prediction on your gene of interest. And I will illustrate this by going to the first tool I will illustrate is the GPI Anchor Prediction tool. And for this, I will go to a gene page in TriTripDB. So let me go up to the top of the page. So in this case, I just went to TriTripDB and typed in either you can type in the ID for the gene up here in the gene ID search, or you can search with a keyword. And then this will return a list of results, and then you can go to the gene of interest. In this case, I chose this gene here, and I'm going to go down and using this left-hand contents menu, I'm going to select the protein features and properties, and this will take me all the way down on the gene page to the protein features and properties section of the gene page. And you will notice here that there are a number of tools. Some of these that were available in our previous release, release 40. The new one I will highlight right now for this particular gene is the GPI Anchor Prediction tool. And for this, we're using a predictor called Big PI Predictor. They have they've trained their data on two training sets. One was specific to protozoa and one was just metazoa in general. And in this case, I'm picking protozoa because we're looking at a trypanosomatid, but often the results are quite similar whether you select protozoa or metazoa. And I would recommend that if you are interested to actually try both of these for a gene of interest if you want to see if it is GPI anchored. Once you select your taxonomic set, you click on Submit Query. This opens up a new page with the prediction made available down here. And so as you scroll down, you'll see this is the amino acid sequence of your gene of interest, the gene we came from. And right here, highlighted in black, there's an amino acid right here. This is the amino acid that is predicted to be GPI anchored, GPI modified. And so this is pretty straightforward and easy. I think one thing as would apply to any tool that you run on genes, any bioinformatic tool, is that you also have to think about it scientifically as a biologist and decide whether this makes sense or does not make sense. For example, proteins that are going to be GPI modified, they typically go into the endoplasmic reticulum. And so if you know your gene is not going there, then that's an indication that maybe this prediction probably does not hold true in real life, basically. The second tool that I would like to highlight is the InterPro Scan tool, and for this I'm going to bounce to PlasmoDB. Again, hopefully you can appreciate that all of our databases have essentially the same functionality and same similar content, basically same structure. And again, I'm going to go down to the protein features and properties section. And for this, I just took our default gene, which is the AMA1 gene. And again, you will see that the GPI Anchor Prediction tool is available here. As I scroll down, you'll notice here there's a table for InterPro domains. So for all of our genomes that we load in EuPathDB databases, we run InterPro Scan by default as part of the genome analysis tools that we run, and we provide the results of InterPro Scan here in a table format. But as well, if you scroll down a little bit more, graphically, we also show these domains in this Genome Browser view for the protein. An important point here is that since these analyses are run at a point in time when the genome was loaded, they could potentially become stale, and that is because InterPro Scan is updated on a regular basis, much faster than we can reload and reanalyze all of our genomes on a release per release basis. And so in order to make it easier for you to see, well, are there other InterPro domains that are predicted based on the most recent InterPro Scan, or for example, sometimes some InterPro domains get, or PFAM domains no longer meet the threshold, and actually InterPro Scan will not predict them to be domains. So you could imagine running an InterPro Scan last year on your protein of interest, and it may have a set of, let's say, 3 domains, but then you run it today again, and you'll find out that, oh, now it has 5 different domains that are predicted, and some of them, some of the ones that were predicted before no longer are there. So it's really important to reanalyze your proteins on a regular basis if you're interested in the domains. But this tool here, which I'm showing with my cursor, is simply an ability to submit your query or your protein of interest to InterPro Scan. Once you click on this, this will open up a new page, which will take you to the InterPro domain page, and it already submits your protein to the analysis, and this starts running. This typically takes a couple of minutes to complete, so I pre-ran this before the webinar. And so for this particular gene, where I clicked on Submit Query, the results appear here. And in this case, there were no surprises. The gene has the same domains that we predicted, but then there are additional domains that are predicted by InterPro Scan that we may not be displaying on our page, and other features that may be of interest to you. So, so these are the 2 new features that we loaded in the current release of EuPathDB. I will now switch over to Suzanne, who will highlight some new data sets in CryptoDB. So Suzanne, I will make you the presenter, and you can show your screen.
Okay, thanks, Omar. You guys should be seeing my screen. All right, so I'm going to scroll down to the news release for CryptoDB 41, and we can see that we, in 41, we released 1 genome sequence, 2 whole genome sequence of isolates datasets, and then 3 RNA sequencing datasets, which is what I'd like to concentrate on today. So these are integrated pre-publication from the Widmer lab at Tufts University. And so all of our RNA sequence data is run through a pipeline that aligns the reads to a reference genome, determines expression values, and then calculates differential expression when appropriate. And then we use those analysis results to build genome-wide searches of these individual data sets. We represent each gene's expression pattern in these data sets on gene record pages. And then we offer dynamic visualization of the data aligned to a reference genome in a GBrowse, right? So those are the 3 basic ways that you can mine this data. And let's take a quick look at each way that you could mine this data. So I'm going to concentrate on this first data set here, the transcriptome of parvum intracellular stages, which is RNA sequencing data taken at 2, 24, and 48 hours post-infection of bovine kidney cells, right? So if I click on the first link here, I go to the record page about this data set. This is a dedicated page and contains information about where this data come from, who are the contacts, if you need to get in touch with people. If there were publications, they would be linked here. There's a description of the data set. And as I roll down, there are example graphs. This is something that you might see on a gene page. And there's a list of searches that are available for this data. Now, because this data, the experimental design included replicates of multiple samples, then we were able to apply statistics and determine differential expression, and we've created a genome-wide search against that. So I'm going to click on this middle search here, and we can run that search, right? So there are several parameters that we need to fill in. There's the experiment, only one choice there, and I'm going to choose the 2-hour time point as a reference and compare that to the 24-hour time point. Look for upregulated genes that are upregulated by a fold difference of 2 or greater, and a adjusted p-value of 0.05 or less, right? Then I can click Get Answer. And I'm taken to a result page. At the top of the result page is a graphic representation of the search I just ran. And at the bottom is our result list. So a list of genes and associated data with that. So our search then returned 220 genes that were upregulated at the 24-hour time point versus the 2-hour time point. And I can see that here in my strategy panel. I can also see that, you know, here or in the organism table. And the result table then includes my list of 220 genes that were returned by the search. Now, you can mine the data in this way by you know, looking at the data here, the fold change value. You can sort each of these columns based on, you know, yeah, you can sort each of these columns. You can also add columns with this button over here on the right, Add Columns button. There's a panel where any of our data that's associated with genes can be added to this table. So this first column here is the gene ID. And that is actually a link to the gene record page. So I'm going to click on that and we can look at the data there. Now, Omar often likes to use the show contents to navigate this page. Since I know I'm going to the transcriptomic table and there's a shortcut up here, I'm going to click at this transcriptomics little shortcut right here, and that will automatically take me to the transcriptomics table. And I have closed all of the experiments, right? So this table is actually, each row is expandable. Each row is about a different data set. And I'm going to search for the data set using a keyword from the title, which shows me to experiments that contain that word in the title, or I don't know, series of letters. And I've opened that row and I can to receive details about this experiment. And here I see that for this gene, there's very little expression at the 2-hour time point, 2,000 FPKM at the 24-hour, and even more, almost a doubling again of expression at the 48-hour time point. There's other data that you can look at on the gene page. If you open up the coverage, you'll see a snapshot of the RNA-seq reads aligned to that. You can open up a description, a text description. You can also add other graphs to the gene page. And you know, you can also consider this in context with the other data sets available for this gene just by opening other rows and mining that data. So the other method of mining this data would be to visualize it in the Genome Browser, and I'm going to use this link here. We're still on the gene page in the transcriptomics section, in the transcript expression table, right? I'm scrolling down to coverage. I've opened that up, and I'm going to click on the View in Genome Browser link, which opens the Genome Browser to a preset location, including your gene, right? I wonder if that's a little big. So, so here we have the annotation track, and this line represents the gene that we were looking at, the disulfide isomerase. And then below that, we have the RNA-seq coverage plots associated with this data set at 2, 24, and 48 hours. And you can see the increase in expression from 0 to 2 to 24 hours. The advantage here at looking at it in GBrowse is that you can actually, zoom in and out. It's, it's a dynamic setup, so you can move around the genome to look at different areas, and you can also use the Select Tracks page to turn on other data. So for example, we can actually turn on some proteomics data and go back to the browser to see that this gene is expressed actually at the protein level. Sound good? Omar or Eve, do you have anything to add to that?
No, this sounds great. I think
okay.
we can move on.
All right, so I'm going to turn it over now to Eve. And she's going to tell us about some of the fungi data.
All right, you should be seeing my screen now. Thank you, Suzanne. Hello, everyone. So I will talk and review a few data sets that we have integrated in the past release. I'm currently on FungiDB news page, and notice that we have several sections dedicated to various types of data. For example, at the top, we have genome sequence annotation that is that appear in FungiDB for the first time, such as new genomes. We also have a section for genome sequence annotation that included updates, and I will review Cryptococcus neoformans KN99 update. We also have a few data sets amongst the transcriptomics, proteomics, and ChipSeek data, and I will go over the proteomics dataset and one of the ChipSeek datasets. We have also integrated variant analysis data for Cryptococcus, Neurospora, Paracoccidioides, and Candida albicans, and updated the phenotype data, which is a combination of curated phenotypes for Neurospora crassa, and include the characterization of the knockout alleles in these fungi. The previous update also included information such as link outs to external databases, and these were some of them were Cryptococcus specific databases such as Phenome or transcription factor databases, and we have also now provide link outs to the peptidase database MARUPS, such as the one shown here, and CBI RAS-seq, and also carbohydrate active enzymes, or KZ database for a selected number of species, but we are planning to expand the list of species for which this information will be available in the future as well. Also just wanted to touch briefly on the section that is called Updated Functional Annotation, and that is essentially a summary of annotation updates that include gene products, Go terms, EC numbers, gene names, whatnot, and also a list of species for which this information has been updated. For example, in the past release, we worked on Coccidioides immitis, Cryptococcus species, and Neurospora crassa. And if you're interested in looking a little bit more specifically at what type of annotation updates were included in each release, click on the Summary of Annotation Updates link, which is already pre-opened in a separate tab. And this Google document essentially includes a detailed and up-to-date information on everything that went in the release, including the number of products and the number of genes that have been updated. And if you want to sort of dive into individual components of the release, you can look, for example, at specific organisms such as coccidioides, where any and all updates are listed according to their classification. For example, GBXref will be link out, and you can also see that we have updated PubMed ID references and other data features. Okay, going back to the FungiDB page, lastly, we have reviewed and curated several user comments, and Amar will touch base on user comments in a little bit in greater details, but I just wanted to mention that the user comments are normally the fastest way to integrate up-to-date information about genes of interest or to showcase your own work. So thank you to all of the users that have submitted user comments, and these particular user comments reflected, for example, updates for gene names, or, for example, functions of genes and various metabolic processes, and etc. Okay, so let's go back to our genomes, and I will go over the updates that we have introduced for Cryptococcus neoformans, or KN99 strain. As Suzanne previously showed, if you click on the link, you'll be redirected to the record page for this particular genome that provides a little bit more information about the data set itself, where the data was loaded from, and if there are any additional nodes from the provider. In this particular update, we have several corrections that were introduced to the gene model. And these were, so the corrections were introduced for gene mappings and functional gene annotation. For example, old locus tags were remapped to new locus tag. And also all gene products were remapped to new locus tag as well. So let me explain you what exactly that was and how do you find the new information on FungiDB record pages. I will go back to the home page and essentially show you how to create a search for genes using our taxonomy option. So if I click on the taxonomy link within the search for genes panel and then click on the organism, I will be redirected to a search page that will ask you for to create sort of a parameter. And that's essentially the organism that you would like to search. In my particular case, I'll just clear all just to get rid of the default settings. And in the search box, I'll enter K99, trying to sort all of the over hundreds of genomes that we have to display one species that I'm interested in only. And when I click on Get Answer, the search will be deployed and essentially will produce a list of all of the genes in this particular genome. And as you can see, this is a very brief strategy, search strategy in FungiDB, and you can expand that strategy by adding steps onto that. So the main output of the strategy, and I'll just close this particular table here, the main output of the strategy is the gene list below. For example, you can click on individual genes to review the information available for any particular gene within this table. And let's take a look at one of the first genes on the list, this guy right here, and I already opened it as a separate tab. So how the information was updated for this particular genome. I noticed that we have updated the functional annotation, which is essentially displayed at the top of the gene record page, to correct the previously misannotated records. However, the gene IDs at the top are still remaining to be old gene IDs. In order to deliver the updated information as fast as we can from GenBank, we have incorporated the updated locus tag or new locus tag within the gene record pages within the annotation, curation, and identifiers section, which is essentially the second content submenu on the left. So if you click on the menu in blue, you will be redirected to the section that also contained a subsection called names, previous identifiers, and aliases. So the gene ID listed within this section is actually the correct and the updated gene ID for KN99. Note that the genome will be reloaded in the subsequent release so that the new gene ID will become essentially the default gene ID and the model will be completely corrected. But while we are reloading the genome, we wanted to make sure that the users have access to the latest information from GenBank. If you're interested in downloading the updated gene IDs, you could navigate to the annotation curation section and essentially click download link here. However, some of you may be interested in essentially exporting information for all of the genes, over 7,000 of them. So that's probably not the best way to approach and download data one by one. There's another way to download this information. If we go back to our search strategy that we just ran before, and I'll close this to display the gene result table. So in order to download the new gene IDs for all genome, click on the download button at the top of the gene results table, and you will be redirected to the interactive screen that essentially allows you to pick and choose the type of data that you would like to export. If you're interested in just getting the mapping of the old gene IDs to the new gene IDs, I would click on the tab delimited, which is the second Excel option form. Navigate to the annotation, curation, and identifiers, and click on names, previous identifiers, and aliases. Note that you have three different types to as a type of export for this data. You can choose to export it as a text file or Excel file, or you can show it in browser. I have pre-ran this search strategy just here, and this is essentially what will be your output if you choose to view this in the browser. So note that the old or the current gene ID is displayed on the left. The new and the correct gene IDs are displayed as aliases, and it's essentially available for all of the genes. If you would like to export more information, such as not only the mapping of the old to new gene IDs, but also an updated gene product information associated with every gene, click on the tab delimited Excel option, which is the first one that allows you to create your custom table. And in this case, I'll just clear all for now. And let's say I would be interested in exporting the product description along with previous IDs. And so again, I will click on show in browser and click on get genes. And I just pre-ran this to save the time. And that's essentially what will be your output, whether it's text, Excel, or view in Gene Browser. Now you have a complete information of all of the updates that have been loaded in the previous release. Okay, so let me go back to our new section and talk a little bit more about the next data set that I would like to walk you through.
And while Eve is doing that, just to highlight to everybody that, again, as I mentioned, since the software and infrastructure is all the same in all of our resources, any tool that we highlight in one of our resources would be the same exact tool that you can run in other resources. So for example, if you were trying to get previous IDs for a Plasmodium genome or a candidoplastid genome, you would follow the same steps that Eve just highlighted.
All right, thanks, Mark. All right, the next dataset that I would like to walk you through is the proteomics dataset that we have loaded for Cryptococcus neoformans JC21. For those that are not fungal specialists, so before I will demonstrate the search, I'll just briefly mention what are the specifics of this dataset. So notice that the authors have looked at the vegetative cells, yeast, and also spores, and essentially looked at the proteomic fractions from the 3 different replicates per each sample. So fungal spores in general, while they're essential for long-term survival in fungi, they're also considered to be an infectious particles that, when, for example, inhaled, can cause temporary or persistent fungal infections, form biofilms inside organs, and sometimes lead to more invasive forms of disease that can be lethal. So looking at the gene expression level, at the proteome level, can be quite useful when trying to understand the processes that occur when fungi respond to different stimuli, such as different types of environments. This particular proteome was extracted from Cryptococcus neoformans, which is a human pathogen. And when I'll walk you through as to how to set up a proteomic search using this data set. So let me go back to home and navigate to our proteomics section within the search for genes panel. It's located here in the bottom. And if you click on the link, it will expand to produce an option. In this case, I'm looking for a mass spec evidence that is linked to the Cryptococcus JC, which will be contained right here. And I'll select only one data set, JC21, and notice that there are 3 replicates for 2 different samples. So in this case, I will choose to look for the protein expression data from spores, replicate 1. There's several parameters that you need to keep in mind when setting up your experiment, such as minimum number of unique peptide sequences. And that's normally sets the minimum number of peptides that are mapped to a gene before you return the search. So requiring more than 1 peptide to map to a gene sometimes helps to eliminate false positive results, but also experiment dependent. So I will leave my settings at default, but you could imagine that after running a search, you may want to go back and tweak your results a little bit more to see how the stringency affects the results and whether the data makes sense when you try to investigate it. So here we are. The screen is refreshed, and I'm returning 18 over essentially slightly under 2,000 genes that are expressed in the spore fraction. My other question was, well, can I look at the genes that are actually expressed in the yeast form, which is non-infections form of Cryptococcus, and then subtract them from those genes that are expressed in the spores, which are considered to be an infectious form of Cryptococcus? And the answer is yes, you can. And you can construct this strategy within FungiDB by essentially clicking on Add Step and navigating through a series of steps that will take you essentially to the proteomics section right here, selecting mass spec evidence, and essentially go through the steps that I have just shown you. Looking for JC21, and in this case, I will take only 1 replicate from the yeast fraction, and scroll down a little bit to the bottom. So these are the essentially the 4 types of search parameters that we can use, such as we can use to intersect, return results as union from both fractions, or we can use essentially these 2 interactive options to return genes that are only present in the spore fraction by subtracting yeast, or the other way around. So because I'm interested in returning only those genes that are uniquely expressed in the spore fractions, I will click on the 1 - 2 parameter and run this step. And I have already pre-run this step, and it's actually faster before I can finish talking, which is great. And that's essentially the your result. So notice that we constructed the strategy by looking at the mass spec data from the spore fractions, yeast fractions, and then we subtracted the genes that are expressed in yeast from those that are in spores. So the returned 384 genes are those that particularly expressed in the infectious form of the pathogen. And from this point, you can choose to download the data, analyze it further, or create even a more elaborate search strategy by using add steps. Okay, so this is it about the proteomics datasets, and please feel free to type your questions in the chat sections. Meanwhile, I'll go back and review one of the ChipSeek experiments that we have integrated as well. The ChipSeek experiment has been integrated for Neurospora crassa and Fusarium, and I will go over the one for Neurospora crassa. So just a brief overview of what exactly we're looking here. Neurospora crassa is a filamentous fungi. It's a model system for a lot of different things, and including also plant biomass degradation. It's been found before that the transcription factors CLR1, 2, and XRL1 play an important role in regulating genes that are important for degradation of the cellulose. And previous experiments demonstrate that there are several genes that are affected. And I would like to take you to the ChipSeek and also compare to the integrated transcriptomics data in FungiDB to demonstrate just that. So one of the genes that has been known to be affected by the CLR1 transcription factor is the CDT2 gene, which is, notice I have already in the search box here. I just quickly search for the CDT2 and I navigate to the search results table at the bottom, and we'll click on the CDT2 gene listed within the results page to be redirected to the gene record page. While it's running, here we go. So Suzanne have previously demonstrated that you can look a little bit more closely at the different types of content within the gene record page, but I'm interested in specifically ChipSeek data, and then to see what kind of other tracks I can upload and compare my data to. So I will open the this gene in Genome Browser, and I have just opened this gene before in the separate tab, and here we are in gbrowse. So on the bottom you will notice that there are several tracks from the ChipSeek experiment, and I loaded this track by navigating to the select tracks tab, clicking on the epigenomics, ChipSeek, Neurospora crassa, and selecting the data set that I was interested in. Going back. And so here you can review the gene model, the annotated UTRs and their location, and also look at the ChipSeek data that have been integrated for one of the samples. So this is a CLR transcription factor that is linked to the green fluorescent protein. And what authors have demonstrated that this particular transcription factor binds upstream of the CDT2 gene, which is a hexose transporter gene. And it has a dramatic effect on its transcription when fungi grow in the presence of cellulose. To prove that, they have also created an RNA-seq data, and in FungiDB, we do have a number of transcriptomics data that you can evaluate these findings against. And to activate other tracks, I would navigate to the select tracks, transcriptomics data, RNA-seq, choose Neurospora crassa as well, and activate the induction of lignocellulose degrading enzymes, which I just did in a separate browser, and I'll show you the output of this now. So notice again on the bottom, we have a ChipSeek where the CLR1 matches or binds rather the gene upstream on the right. And these are the RNA-seq coverage plots from a separate experiment where wild-type cells were grown in the presence of various types of media, including those of cellulose. And notice that there is a dramatic increase in the gene transcription, which is consistent with the data that has been found by the authors here and goes hand in hand with the sort of experimental data in FungiDB. So that's one of the ways how you can take tracks, whether they're RNA-seq or ChipSeek, and overlay them with integrated data in FungiDB to sort of create in-depth investigation of the genome or specific genes. All right. So this concludes the section for the ChipSeek. Let me go back, and I will show you one more search, and that's concerning the phenotype data that we have updated in FungiDB as well. So notice that phenotype data, again, because it's a search for genes, can be found within the section shown here. And if you click on phenotype evidence, you will be redirected to a table that lists the various types of phenotype records available in FungiDB, as well as the genomes for which these records with which these records are associated. In this particular case, I will review the phenotypic analysis data that was shared with us pre-publication by Catherine Berkovich, and this is a curated data where the authors have taken a knockout mutants, have grown them on different types of media, and have looked at the phenotype that was associated with various developmental stages of the filamentous fungus. So in this particular case, I'll scroll down and let's take a look at the ascospore number production, let's say, for example. Just a quick introduction into ascospores. So these are essentially spores that are produced by mating of two strains of opposite mating types in the filamentous fungi. And the ascospores normally have four meiotic processes, four meiotic products that are then divided mitotically to produce 8 ascospores in one ascus. And normally, when you have gene knockouts that affect the ascospore production, it means that there is an effect on some sort of either a meiotic process or the process that is required to complete the mating process correctly. So in this case, I'm interested to look at any gene knockout that was associated with reduced production of spores. And I would choose reduced and severely reduced. And I understand that there's, I noticed that there's 2 separate sections for the severely reduced, but it's just the way algorithm have recognized reduced. The capital letter is being treated as a separate metadata entry, and this is just a small bug that will be addressed in the next release. But let's go ahead and select everything that has a reduced impact on ascospore production, and I'll click on Get Enter. And our search will return over 70 genes that match the specified criteria. Imagine that you could further evaluate those genes not only for the type of the product that is being produced, such as where this may play a role within the meiotic process, but you could also overlay these particular results with other data within FungiDB, such as another phenotype that may be associated with this particular genes as well. This information can be particularly useful when studying gene knockout mutants and trying to understand either metabolic processes or mating processes or any deficiencies that mutants experience, whether biological or cell function, etc. All right, I think this is all on my end, and I will pass it over to Amar, who will talk to you briefly about the user comments and how you can develop the annotation with user comments.
Okay, thanks, Eve. Let me see if I can change the control. I'm making myself the presenter and show my screen. So I'm still looking at FungiDB here, and one thing to note is that up here it says my name and my profile. That's because I logged in. And there's while most of the functionalities available in EuPathDB databases are freely available, well, everything is free, but most do not require you to log in. Some of them require a login because they're associated with your profile. And one of these features is the ability to add a user comment on any gene page on any of the databases. And to add a user comment, and I will also explain why these are useful, but to add a user comment on any gene page, you will notice that there's a little section here that's called add the first user comment. If there was already a user comment here, it'll tell you how many user comments are available, and those are all available in the annotation and curation section of the gene page. And as you scroll down, it'll tell you if there's any information. In this case, I picked a gene that is hypothetical, at least its function is hypothetical. There's not a lot known about it, but let's say this is a gene that either you have worked on and you know its function, or you've read about this in a paper where its function has been published or determined. And so you can go ahead and add that information on the gene page without having to wait for until the databases are updated with a newer version of the annotation. So to add a user comment, you can just simply click on add the first user comment. Again, you have to be logged in. There are only a couple of things that are required. So for example, I can say the headline, this is a kinase. Let's say I've determined this. And then you can say, based on, lab assays, and then see publication. And then as I scroll down, you'll notice here that there's a section to add a PubMed ID. And I can I'm just randomly adding a number, but you can quickly click on preview and make sure that you have the right publication associated with it. Obviously, I picked a random number here, so it's like playing the lottery. But typically, you would obviously add the PubMed ID that's associated with your evidence. And there are other things you can add. For example, I can upload a file. Let's say you have a image subcellular localization or a kinase assay showing the results of the kinase activity. I can add that here, and I can also associate my user comment with multiple genes. Let's say you're trying to provide the same information about a gene family or a secondary metabolic pathway or whatever else. And then you simply click on add comment. I'm not going to do that right now because we don't want to add this comment on this gene page. But as soon as you click on add comment, the comment actually becomes immediately available on the gene page in the comment section, and it becomes searchable via the text search. So for example, people will be able to search by your last name or your name because you are the submitter, and then find all the genes that have your name associated with them. Or they can search based on any keyword that's in your headline or in your comment. You could also search the entire database for any gene that has a user comment on it. So under the annotation and curation and identifiers section, I can select user comments and then say get answer. And this will return all genes. In this case, I'm looking in FungiDB. It tells me there are 2,017 genes that have user comments. And as you scroll down, you'll start seeing that these are the genes that have user comments. And if you want to get more details about them, I can click on any one of them. And you'll notice now when this page loads, it says, oh, this gene has 5 user comments. So if I click on this, it'll take me to the section with the user comments. And once they load, it will have a listing of all the user comments, including who added them and some additional information that populates this table. And then you can go in and look at the details of each comment. And so if you click on the comment ID, then it shows you all the comments with the information about those comments. So a user comment can actually be as simple as saying that I tried knocking this gene out and it did not work, right? This is very useful information. And you can imagine any information you would have liked to know about a gene when you visited the gene page, but wasn't available, but would have been nice to have available as a user comment. That's really useful information, and I would encourage all of you to go ahead and add those user comments. Besides, the comments appearing immediately on the gene page, for many genomes we have curators who actually are updating the gene annotation. This is specifically for Plasmodium genomes, some of the canidoplastid genomes, and some of the fungal genomes. And all of these user comments get automatically submitted and forwarded to our curators, or the curators from Sanger, and they take a look at them and determine and evaluate whether that comment warrants a change in the annotation. And then they would actually go in and make the change as appropriate, and that will appear then in a future release in EuPathDB once we reload the genome. But the user comment is a really nice and quick way for you to add your information on a gene page without having to wait for a curation to occur. Or if your genome does not have ongoing curation, it's a very nice community way for people to add their information about genomes, very low throughput way, and actually quite cheap. Okay, so moving down, we have, as you can see, there are a number of different data sets that have been loaded for all of our releases. Eve just covered FungiDB. We also loaded a few genomes in MicroSporidiaDB. So there were 3 genomes, and we also loaded some population data, which you can query by and look for polymorphisms in genes based on the samples you select and the sample information that's available to us. And I will switch very quickly or soon to Suzanne, and she will actually run through one of the searches on polymorphisms in PlasmoDB. But before that, just one quick note is that you'll notice that some of our databases, it says, there's no new data. And that's often the case. So not all of our databases get new data in every single release. It's just there are too many genomes to do that. But we do try and get data for all of our databases a couple of times during the year. So if your favorite organism did not get data this release, the next release, they may actually get data. Okay, I will switch over to Suzanne, and she will cover polymorphism data in PlasmoDB. So Suzanne, I will change this to make you the presenter. And as a reminder, again, feel free to ask questions or add comments as we go along.
Okay, thanks, Omar. So here I am on the aggregate news page for EuPathDB, and I'm looking at PlasmoDB 41 release. And the first bunch of data sets that come up are genome sequence and annotation for 15 isolates, right? This is data from the PF3K malaria gen project, and there is an associated publication. The data for these genome sequence and annotation data sets are PacBio and Illumina. So these are long read assemblies, and they carry all the advantages of that. Several of these are lab strains, right? 7G8, DD2. So if you're interested in looking at the annotation in our system for these, you can go ahead and do that now. In this data set was also PF3D7, but we did not integrate the sequence and annotation from this version of PF3D7. We retained the community accepted as a reference. Okay, so for these genomes, not only did we integrate the genome and annotation, but we put the Illumina data through our genetic variation pipeline. And that is this data set here. And I'm going to click on this link here and go to the record page associated with this data set. And of course, there's the publication is linked up top. There are contacts and a description of all the how the data was collected. And then here under searches that use the data, we see we have 8 or 10 searches available having to do with SNPs, right? We can look for SNPs. We can look for genes associated with the variation that we learned about from these whole genome data sets. So I'm going to demo this search right here, identify SNPs by differences within a group of isolates. So I click on that and I go to the record page, and I'm going to go ahead and scroll down to the help section that is beneath the search. Every search page has a help section or description below it, just to talk a little bit about how we deal with this data. So first, when we have whole genome sequencing of an isolate, we align each isolate's read to the reference genome. So, and these blue lines here represent reads from a single isolate, and we align them to the genome and then walk down the genome base by base and call SNPs, right? And then that allows us to compare single nucleotide polymorphisms across isolates, and we build searches so that you can find SNPs that are in common to a group of isolates or distinguish between two groups of isolates. The search I'm going to demo for you now is to look for SNPs based on differences within a group of isolates. The search opens with default parameter set, and we are looking at PF3D7 isolates, or isolates that were aligned to 3D7. There are a total of 208 isolates in our system, and through this metadata panel here, we can see, we can look at all of the data sets that these 218 are distributed through. And I'm going to click here on the data set that we were just looking at, geographically dispersed p-falciparum isolates, the PF3 3K data that we were looking at. So I'm going to look for isolates within this group of 16 isolates and leave these as default settings and click Get Answer. So you can see that my search returns, a ton of SNPs, 190,000 SNPs, and at the bottom, the meat of the results here is a list of records associated with each of those SNPs. But I can go back then and actually make this search a little bit more stringent by increasing some of the parameters here. So the minor allele frequency I'm going to set to 50%, and the percent of isolates with that base call to 80%. And if you're wondering about what those mean, you can come down here to the search description and read that. And we also, in these little question marks, define each of the parameters here. So I'm going to go ahead and click Get Answer. And we see that we've reduced those 190,000 SNPs to 670, right? So our results table then is a list of SNP IDs returned by the search and then associated data, right? And when a SNP is located within a gene, you're given a column and a link to that gene ID, its position in the protein, whether that affects the protein sequence or not. So let's click this link here and go to the SNP record page, and we can look at that. It's just like any of our other record pages. It has summary data at the top, interesting information about what the major and minor alleles are, and sequence surrounding the SNP so that you can take that to another program if you want to. And then as I scroll down the page, I see other summary information about this SNP in relation to all of the isolates, right? We searched our 16 isolates to come up with this SNP, but then we can look at the data associated with all of the isolates. We can also, so under the SNPs alignment category, we can run an alignment to look at that a little bit closer and choose individual isolates to run our alignment against. I'm going to go back to my results by clicking on this My Strategy tab here, and I'm going to look at this gene ID column here. So this SNP is actually within this gene, and so I can go back to that gene page and look at the SNP information there. I use this shortcut here to go to the SNP section. And again, it's going to show us a summary of you know, an image with all of the SNPs that we've called against this gene that occur in this within this gene, and then summary information. Another possibility to produce an alignment down here. So there are other interesting, just one more, I'm going to go back to the home page. One interesting search, well, I think I'm going to stop here. I think that's it. That's an overview of the data from about SNPs that was released in 41 PlasmaDB.
Okay, thanks,
Okay.
Suzanne, and I'll go ahead and switch it to me. Moving down, so in addition to the SNP data and the genomes that Suzanne mentioned that we've loaded in the most recent release of PlasmaDB, there have also been a number of annotation updates which come from the curator at GeneDB and Sanger. And those are listed here in a table, just like Eve indicated for FungiDB. You can go in and take a look at these. And then we hopefully integrate these then at a future date when we update the load of the genome, and then they appear on the gene page. But we do have immediate information about what has changed and allows you to go in and look and find out if your specific gene of interest happens to be on that list, or if there are new annotations that you may be interested in. Okay, moving down, ToxoDB received 2 new datasets in this release. One of them was a microarray dataset, which I will describe briefly, and then the other one was simply a sequence from a clone of Toxoplasma, which is available for alignments and SNP calling, just like Suzanne showed you. For this microarray dataset, so this specific, again, if you click on the title of the dataset, it takes you to the dataset page, which has additional information, including a link to the publication. In this case, the researchers from Kami Kim's group deleted the methyltransferase gene to try and understand its function. Again, we loaded the expression values for the microarray results. There are 2 searches that you can run on this data. One is a fold change search, and the other one is a percentile search. And again, I'll just go ahead and demonstrate the fold change search, which will look identical to the search that Eve showed you earlier on with RNA-seq data. Okay, so essentially, you can go in and ask for genes that are differentially regulated between the samples in the experiment. And in this case, I'm going to ask for genes that are downregulated in the knockout compared to the wild type and the complemented strain. And I can change the parameter here. So let's go ahead and select, we want to look for genes that are downregulated in this case. And I want to compare the minimum expression value from the 2 samples I selected in my reference samples compared to the expression value in the knockout. So basically I'm saying, and I'm keeping the twofold difference here as a default. So I'm basically saying, give me all genes that have a twofold reduction in expression when I compare the knockout to either the wild type or the complemented strain. So this graphic here gives you an indication that you selected the right parameters that you're interested in. Again, click on Get Answer. This will run the search and return a search strategy that includes the results. You can see I ran this search before as well to make sure everything is working. So I'm just going to close this. It's essentially a duplicate of this. So there were 10 genes that were returned that were downregulated in my knockout compared to my wild type and complemented. And one thing you want to immediately do, of course, is see if the gene itself that was knocked out is also in the list of genes that are downregulated. And as you scroll down, you will notice that here it is. So PRMT1 is here. It is downregulated in the knockout compared to both the complemented and the wild type. So that's a good thing, right? If it's not in this list, this would raise an alarm bell, at least for you to take a look and question whether the results were loaded properly or there's an issue or something else that you need to understand about the experiment. Okay, so going back to EuPathDB, I'm going to move down to TriTripDB. So TriTripDB received, again, a number of different datasets, including a few RNA sequence datasets, quantitative proteomics dataset, and a 5' start site dataset. I'm going to quickly run through a couple of these datasets, and then we'll turn it over to Suzanne to tell us about the start sites, and then we will be towards the end of our webinar. For the transcriptomic datasets, really, again, not to replicate all the searches we've been running when it comes to fold change values, whether it is transcriptomics or microarray datasets. These you'll run essentially the same type of search, but to highlight here that some of these experiments are both parasite and host response data. And you can search the parasite data in TriTripDB, but also we have another database called HostDB, and you'll notice here that there's a link to HostDB and a dataset in HostDB. So I can open this. This will take me to HostDB, again, to the dataset page in HostDB, and this shows me, again, this experiment, which paper it came from. And I can go down and say, well, I want to look for genes that are differentially regulated. Let's pick this differential expression experiment. Again, Suzanne demoed this earlier. I think Suzanne or Eve, but the bottom line, Suzanne did. So the bottom line is you can compare two samples to each other. So for example, I can say, well, I want to compare the human fibroblast cells 96 hours post-infection and compare them to the uninfected cells. I want to look for genes that are upregulated by at least twofold. And again, as Suzanne did, I can change the p-value to 0.05 and then click on Get Answer. And this will return all genes that are upregulated later during the infection. And you'll notice here I got almost 2,600 genes, which seems like a huge number of genes. And you may decide, well, this is really a bit too lenient. I want to revise this step. I can always click on the Edit function, click on Revise. It reloads the page with all the parameters I selected, and now I can, for example, change the fold change to, let's say, 10. Right? So you want things that have even a higher fold change, or I can change the p-value and make it even more stringent. Right? And so this will return all the genes that are that were upregulated in my infected cells compared to my uninfected cells. And again, we're looking here at human genes that are upregulated in response to an infection with T. cruzi. The other data set from TriTripDB that I will highlight is this quantitative proteomic expression data set. So just as we highlighted earlier that we load peptide data, so you can go in and ask for genes that have a certain number of peptides from a particular sample from an experiment. But that is not quantitative, it's semi-quantitative. So what we do also load is quantitative data. In this case, it's a quantitative proteomics experiment looking at the developmental cycle in T. brucei. And so again, information about experiment, there's an example graph. This is a gene that's upregulated in procyclic forms compared to developmental stages all the way from long slenders all the way through procyclics. And I can go in and actually run a search. And hopefully the search now is looking familiar to you. So this is a similar search as we have for all of our fold change experiments. But I can ask for genes that are upregulated in, let's say, my procyclics and these later time points in development compared to the early time points, and ask for genes that are upregulated. Again, I can change the parameters here to, let's change this to minimum and maximum. Here we go, and maximum. Okay, so I'm looking for genes that are upregulated in my red samples to my blue samples, and I'm asking for a 1.5 fold change between them. and then you run this, and again, you will get the same type of result, the list of genes that met the criteria of my search. You can look at the genes, you can look at their look at the graph of expression and confirm, okay, this is the type of gene I was looking for. There's some quick analyses that you can run on your results. For example, I can click on this little graphic here. This will create a word cloud based on the words in the names of the genes. You'll notice the first few words are not terribly useful, so I can actually eliminate those from my word cloud by simply changing the number of genes that show up. Okay, let's see. There we go. And then I can sort this, and then this will give you sort of a quick view of how often a particular word appeared in your description. So I could see mitochondrial, for example, appeared 6 times. When you mouse over it, a little number shows up. This is obviously not statistical. It's simply a nice, quick and dirty way for you to see if there's anything interesting in your list of genes. If you want to statistically determine enrichment, you can click on Analyze Results. There's a tab right here for any set of results you get. And then you can run any of the enrichment analyses that we have. And these are actual statistical enrichments. So for example, I can look for Go enrichment by clicking on this Go enrichment option. This will provide me with the Go enrichment parameters that I can modify. And once this loads, let's give it a second. There we go. So I can select which Go ontology I want to use in my enrichment. And so let's keep it a biological process. I can play around with the p-value, click on Submit. And so this will determine, based on my list of 79 genes, is there anything there enriched compared to the background, which is the genome itself. And it's using gene ontology as as what's being enriched, basically, or looking for enrichment. The answer may not be anything significant, right? But this is part of the discovery tools that are available to you, and allows you to actually determine if there's something worth pursuing or looking at further in your results. And sometimes, obviously, this takes a few seconds longer, but you can keep this running while you do other things on the database, and then come back and take a look at it. Okay, so here are the results, and so you can see that there's, oxidation reduction process seems to be a little bit enriched. It's good to look at these p-values, including the corrected p-values, to make sure that you trust the results and you think this is something worth investigating further. Okay, I'm going to switch back quickly to Suzanne, and she will tell us about transcription start sites, and then we will wrap up the webinar. So go ahead, Suzanne, you should be presenter now.
Okay, thanks. So so this last data set here, the transcription start sites, is just a GBrowse track. There's no search associated with that. And so, as you may know or may not, that kinetoplastids do not produce transcripts, but with the usual eukaryotic methods, right? They produce polycistronic units that then need to be broken down into individual transcripts. So defining transcription start sites requires, some creativity and alternative methods. And in this case, the Segal lab isolated small RNAs, less than 200 nucleotides, and broke that sample into 2, right? So now they have 2 different samples. They replaced, in one of those samples, replaced the 5' end cap with adapters. And then they sequenced those 2 samples, and those transcripts that had the 5' end cap did not get sequenced. Right? They didn't have adapters, they did not get sequenced. And then they analyze that data and look for the extra bits of sequence that were associated with the treated samples. And then we express that as a ratio and show that in a GBrowse sample, in a GBrowse track. So I'm going to go ahead and click on this link here, View Data in GBrowse. This is the kind of the easiest way to get to that data. That may take a while to load, so I actually have it already open here. And what that link is a pre-configured I don't know, track configuration within gbrowse associated with the with that data set, and it opens with the annotation track on the top, our transcription start sites are next, and then some ChipSeek data also associated with this data set. And what you can see is that, like, so say for this transcript here, this data set shows a peak at the 5' end of that, indicating that this is where a transcription would be initiated from. The same with this reverse-facing gene. This gene then is associated with this peak in the transcription start site track that indicates this is where transcription has started from. I feel like that's an overview. Is there anything that you would like to add, Omar or Eve, or are there any questions?
No, I think that's good. Let's go ahead and wrap it up. It's almost close to an hour and a half, which is the duration of this webinar. So I wanted to highlight a few things before we end the webinar. One is just to let everybody on the call know and tell your colleagues that we do have a couple of workshops that are registration is open for them. If you're a fungal biologist, there's a fungal workshop, fungal pathogen genomics course that will be held in Hingston from May 7th to the 12th. The deadline is not until February, but it's coming up soon. And this workshop highlights a number of different fungal informatics resources, including FungiDB, but also Ensemble Fungi, Pumbase, SGD, and Microcosm, JGI. It's a full week workshop, and we cover various aspects of these resources, how to use them, how to maximize the utility of them. It's quite a nice workshop. So if you are a fungal biologist and are interested, please go ahead and apply before the deadline. Or if you have additional questions about this, send us an email. Always, click on the Contact Us link and ask us questions. The second workshop is our 14th annual EuPathDB workshop. This is held in the University of Georgia in Athens, Georgia. Again, this workshop is held in June. Deadline is also in February. But it will cover almost all of the EuPathDB resources. It's specific to EuPathDB, and we go in depth into all the different tools and analyses and methods that you can use in the databases, and also quite a popular workshop and very useful. And I would encourage you to apply if you work on any of these organisms. So I wanted to quickly end by highlighting that again and remind everybody that when we choose datasets for loading, this is based on a number of criteria, including, and most importantly, what we hear from the community. And we hear from the community either, when we travel to conferences and meet you at conferences, and you tell us about different datasets. But also we have advisory groups for each of our most of our organism databases, where they guide us and let us know about priorities for a specific organism group. And we schedule our datasets on a bi-monthly release cycle. So every 2 months we have a new release, and they're usually well over 30 or 40 or more datasets that are being loaded for each release. And just to give you a glimpse, I'm going to switch to our internal tracking system. And you'll see here, this is for release 42, which is coming up scheduled, or the target scheduled date is February 28th. And you will see here the number of datasets. I'm not going to highlight specific ones, but just to show you that we'll be loading a number of ChipSeek datasets for Plasmodium and Leishmania. So various Plasmodium species and also Leishmania. We have a number of updates, functional genomic updates for fungal organisms and Leishmania. A number of new genomes, both in FungiDB, in HostDB. We're loading the cow genome, believe it or not, but that's because it's a host for Cryptosporidium, and we have a Cryptosporidium host response data in cow, and so that'll be available in HostDB. We have new genomes for Babesia in PyroPlasmaDB, and Plasmodium as well. Again, a number of trypanosomatid genomes for T. cruzi. If you're interested, we're loading some recent genomes, the DM28C, which is a PacBio, and the TCC, also a PacBio data set from the same group. Those will be available very soon, and there are a number of fungal genomes. Moving down, there's some updates to our Go annotation. We're loading a number of manually curated or experimentally generated phenotype data. So the TripTag data, for example, is subcellular localization of thousands of genes in T. brucei, and those are available with the images of the genes. This data is currently available. We're just loading an update to this. There's some metabolomics data that's will be loaded or is being worked on. Antibody microarray dataset if you're interested in immunity against Plasmodium. This looks for potential antigens in Plasmodium. A number of proteomics datasets. In particular, if you're interested in targeting to the apicoplast in Plasmodium, we're loading some information from a new study that looked at proteins that are targeted to the apicoplast. A number of RNA sequence datasets, as you can see here. We don't have to go through the details of them, but suffice it to say there are many, and from many different experiments, and also a number of polymorphism datasets from various organisms, including Leishmania, Plasmodium vivax-like organisms, and also fungal organisms such as Candida auris or Paracoccidioides. So many data sets. Some of these data sets may not actually make it in the end for a number of reasons. One reason may be that there's a lot more work that needs to be done. And so for every single data set that we load, we communicate with the people who actually generated the data. We show them what we did with the data. They get a chance to take a look at it in a what we call a QA site or a quality assurance site, that they can take a look and they can examine the data. And sometimes there's additional work that we have to do, which means we need to move it to the following release. But in most cases, we release all the data sets that are unscheduled, and we only release them, of course, once the provider of the data or the people who generated data are happy with the way we represent it and are satisfied that it represents the work that they performed. Okay, so we are at the end of our webinar, and I will like to take this opportunity again. If there are any questions that you didn't get a chance to ask during the webinar, feel free to send us an email, and we typically try and respond within 48 hours of the email. If you have any other questions about anything in the resource, tools or functionalities, let us know. Or if you have suggestions for how we can improve things, again, we'd love to hear from you. And besides that, thanks for joining this webinar. The recording of this webinar will be made available in our YouTube channel. And you can, if you are interested, you can access our YouTube channel from any of our pages. At the top right-hand side, there's a link to YouTube, and we have a number of videos there, tutorials that you can follow. And I would encourage you to visit that page. But this webinar will be available there, so you can see it again later or show it to your colleagues if they're interested. All right, so Yves, Suzanne, if there aren't any other comments, I think we can call this call the end of this webinar. So thanks, everybody.
Sounds good. Thank you, everyone, for joining.
Thank you.
Bye-bye.